All right, well, thank you so much for trying to learn a little bit about all things DNA and RNA. All right, so hopefully you've taken some time to look at this slideshow. But yes, there's a picture of ATP synthase that's in the inner membrane of your mitochondria. Acid will diffuse across that membrane and cause this enzyme to make ATP, I guess. And really that's all part of cell respiration. That is a catabolic process. Yeah, remember cell respiration, here's that pathway picture that we've looked at. But yeah, just think about it, it's breaking down sugars systematically in the cytoplasm and in the mitochondria. Now, if we start to do anabolic reactions, we're going to use that energy that was made in the catabolic reactions for that to happen. So really, as we start looking at these pathways in our cell, yeah, I guess if we're not eating and breathing, you don't stay alive, so none of these anabolic pathways are possible. But yeah, just think of these energy molecules as leaving the mitochondria. And they are going to be used to make DNA and RNA. And those structures are built in the nucleus. Okay, so yeah, I think looking at this really helps. It's easy to see how ATP could be used to make RNA. If it's used to make DNA, it's really just called D-ATP. But yeah, it's just that sugar would be different if it was deoxyribose. There's no oxygen there. You can see the phosphate groups bonded together, our five-sided pentose, and then our nitrogen base. So yeah, if we were, so what is adenosine in ATP? Adenosine is adenine and ribose. ATP is adenine, ribose, and phosphate. GTP would be guanine, would be the nitrogen base attached there. So, and once again, just be familiar with how to identify if the pentose is ribose or if it's deoxyribose. Look at carbon 2 right there. Okay. Yeah, so I think most people did pretty well looking at these. It's pretty straightforward looking at my pictures here. But yeah, just please try to remember here, catabolic reactions mean it's breaking down, and that happens automatically. We don't need any outside influence. Things that break down spontaneously break down automatically. All right, when we break down these things, we break down our food, we make carbon dioxide, we make water, you're always breathing those out. Really, as we break down protein, you make ammonia too, and we'll kind of look at how our body handles that. But yeah, once we get through this central dogma, we're going to spend a few weeks just on proteins. Okay, where are we here? Slide. Yes, lipids, remember they're made of fatty acids. They break down into fatty acids. Carbohydrates, they're made of monomers of monosaccharides. They break down into monosaccharides. Proteins are made of amino acids. They break down into amino acids. There's pathways in your cells for all of these things to be converted to pyruvate. Remember, if you go into oxygen debt, it's going to reach an equilibrium with lactic acid or lactate. As long as you keep getting enough oxygen supplied, the pyruvate turns into acetyl-CoA. It may enter the Krebs cycle. But really, acetyl-CoA and these acetates really can also be used to build more things. And that's the whole thing, these anabolic pathways, you really are building things. And now to build this, it takes energy, and that energy comes from this breakdown of ATP. So as we study proteins, as we study enzymes, we'll do a few days or a couple days on thermodynamics. But yeah, really just to put it back together, it takes energy. That energy comes from the breakdown of ATP. But yeah, these are all the different things we're building in your cells all the time. And it's kind of interesting that that kind of broken up piece of sugar with an enzyme, acetyl-CoA, really can enter pathways to become all these different molecules. Now, really the ones that we're focusing on are nucleic acids, DNA, RNA, and protein. Those are the Okay, once again, thank you so much for your time here. Yeah, it helps me remember these processes by remembering where they happen, but replication in the nucleus, trying to copy all of the DNA, transcription, just trying to copy or replicate a small piece of the DNA, really you make it into an RNA so it can float around in the nucleus. Translation is the same thing as protein synthesis. Okay, where do they happen? Replication and transcription. You need DNA for those reactions to happen. That's going to happen in the nucleus. Translation, really anywhere. Anywhere out here in the cell. Anywhere a protein is needed. Okay. So there's another picture. Replication, transcription, nucleus, translation out in the cytoplasm. All right, please, we drew these in class. If you can draw the 1, 2, 3, 4, if you can draw the 5-sided sugar and understand our convention for numbering those carbon atoms, then you can really understand all of our ways we model the genes and model our DNA. Yes, please know the nitrogen base is attached to carbon 1. Carbon 2 is how you can tell what type of sugar it is, whether it's ribose or deoxyribose. And then really at 3 and 5, that's where the phosphate ions will attach. Okay. So yes, if you missed those drawings in class, here they are available for you, but let me know if you need a better copy. There's more links on the website below of these same drawings. So yeah, when we draw the whole thing, I guess just kind of try to remember that hydrogen bonds hold the nitrogen bases together. Phosphodiester bonds maintain the integrity of that backbone or hold the sugars and phosphates together. You can also see from this drawing how we label one side 5 to 3, 5 to 3, 5 to 3. The other side is complementary or anti-parallel, and it's labeled 3 to 5, 3 to 5. And once again, that's just from counting the carbons and the way we number them in the sugar molecule. All right, hopefully you've studied this slide. This is from our National Institutes of Health. Really the same pictures, I believe, kind of show up in the Nature article. But yeah, just different ways to represent DNA and RNA. You can see the structure of all the nitrogen bases there. And once again, keep in mind the hydrogen bonds hold those nitrogen bases together in the middle. You can break those hydrogen bonds and really not fundamentally change what the DNA is. Just changing how those nitrogen bases are stuck together, but they'll still keep the same sequence. The sugars and phosphates will still stay bonded together. Hydrogen bond is not a covalent bond. The phosphodiester bond is a covalent bond, though. All right, so let's take these models that you've seen, hopefully before this class, and look at maybe this is something you've never seen. In a lot of ways, we're very lazy with this. So they're just saying, okay, piece of DNA, there it is, piece of DNA. Let's start labeling some pieces of it. They label some pieces of it with letters. They label some pieces of it with numbers. Altogether, this little thing here, this gene, has 7,700 nitrogen bases in there. Actually, there's the base pairs. So yeah, it's double-stranded DNA. So here's a little piece with 7,700 base pairs. And this is a gene that makes the albumin that shows up in an egg. Okay, albumin's a protein. All right, this DNA, the enzymes come in. The enzymes allow the transcription to happen. So here's the piece of RNA. Keep the same letters, keep the same numbers there. I guess we can kind of assume it probably is close to the same number of base pairs. Then all of a sudden, all these yellows, the A's, the B, C, D, E, F, G, those are gone, cut out. They don't show up down here. So we went from 7,700 nitrogen bases to 1,872 nucleotides, which one nucleotide will have All right, once again, I can't thank you enough for your time. So we were looking at this slide here that tries to model how DNA with 7,700 base pairs gets transcribed, then the primary transcript gets the introns removed, the exons spliced back together. You get what's called a poly A tail stuck on the 3' end. You get a what's called a G cap or a guanine cap, kind of a GTP cap stuck on the 5' end. But yeah, your final RNA is much smaller than the original DNA it was transcribed from. Yeah, now this is the model of how we make albumin, which is a protein that shows up in eggs, and including human eggs. And it's a very, very important protein. It really kind of regulates our blood pressure. And yeah, we'll just show up. We'll talk about it a lot throughout this course. Okay, I don't know if you've never heard of an RFLP, a RefFlip, a restriction fragment length polymorphism. Really what that, if you kind of look at this is supposed to be a chromosome. Do chromosomes really look like that? No. They're not nearly that straight. They don't have perfect lines. They're not that symmetrical. But you can see this banding pattern with the stains that we use. But anyway, going back to what this is trying to show, outside of these, I guess the bands are really where the genes are, or where we got the right stains to stick at least. Outside of those genes are these non-coding regions, but they really vary from person to person. So the value there is if we can figure out the structure of those non-coding regions, it helps us identify an unknown person, whether it's someone who left blood at a crime scene or DNA at a crime scene, or if you're trying to figure out who the father is. Okay. All right, once again, I love this picture, so hopefully you've looked at this and this kind of makes sense to you. We can see the cell, we can see a nucleus, we can see chromosomes. You can even see, portions or pieces of a chromosome with our light microscope. But you're not going to see individual proteins, histones. You're not going to see the double helix. Remember, we pretty much stripped all this DNA off of the chromosomes from the kiwi. Kiwi have chromosomes. And it just was that big snotty mess of goo as we pulled it out of each and every one of those millions of cells that were in the kiwi. Okay. Yes, now the model for replication. If you still have trouble understanding leading and lagging strands, let me know. Essentially, if you remember that polymerase enzymes read at 3 to 5, and it's really working towards the bubble would be the leading strand. So if you look at this picture here, the blue is the new strand being built, and it starts, or it reads at the 3, so the new one is built 5 to 3. So here's the new blue one being built, and it's the leading strand works towards the fork, towards the replication bubble. That little replication fork or bubble was created by helicase, which breaks the hydrogen bonds essentially. Primase kind of helps give it a place to start. So really there could be primase showing up down here too. All right, now if you look to build the lagging strand, it works away from the fork, away from the bubble. And it's still red 3 to 5, built 5 to 3, but as you build this lagging strand 5 to 3, it works away from the bubble. As a result, we get these fragments, and those fragments have to be ligated or joined back together with ligase. Quite honestly, even the leading strand has to be kind of joined or ligated in spots as well. Okay, now we were just looking at this picture of replication, and yeah, we were just looking at this picture of replication. Now, we're going through the different enzymes, but let me show you the picture we made on our board. If you can draw this, then you pretty much got it. But yeah, just kind of randomly labeled one side 5 to 3, which means the new strand is built towards that bubble, 5 to 3. All right, yeah, now if this was a picture of transcription, it would just be this one piece here. So if we were to transcribe RNA, the DNA would unravel, the RNA would be built here, and so this little green piece, this new strand, this what is the leading strand on DNA, if it were made with ribose, it would be RNA, and it could float around and get out of the nucleus and out into the cell. So once again, the lagging strands work away from that replication fork, away from that bubble. And then, yeah, we also asked you to understand the different enzymes that are involved. And really, once again, just think of them as classes of enzymes: helicase, primase, polymerases, ligases, isomerases, or a lot of times they're called topo or topoisomerases. Yeah, and here's that picture from the enzymes from 1982, all right, and we had started to identify them. Look how big these things are. The molecular weight is 900,000. Are you kidding me? I mean, it's really just kind of an estimate. We really don't know exactly how big or exactly what that structure is. But yeah, here's a lot of the proteins that enzymes we found that need to be there in order for replication to happen. All right, so replication happening in the nucleus, transcription happening in the nucleus. All right, and really it's pretty much the same. As far as our purposes are concerned, it's just a different set of enzymes, different set of polymerase enzymes. Instead of the whole genome, it's a smaller single strand. And you don't get thousands of bubbles opened up simultaneously everywhere. You just get the one little bubble and it closes at the end after you've made that RNA. But now there's more to it though, because after it's transcribed, here in this picture they call it pre-mRNA. A lot of books will call it the primary transcript, and then it gets processed and really stuff chopped up, a couple things added to the end, and now you have a mature RNA. But that's still that RNA processing, still part of transcription, really kind of happens in the nucleus still. So yes, if we look at slide 25, here's another picture. We went through this in an earlier screencast. But yes, you have your DNA, there it is, the primary transcript, there it is, it's cut, it's cleaved, stuff is removed, it's spliced back together. And then once again at the 3' end there's a lot of adenine. At the 5' end, this cap is a lot of guanine, a lot of GTP. Now keep in mind this whole processing and cleaving and chopping and shortening of, so from the primary transcript to the mature mRNA, it's a lot smaller. That only happens in eukaryotes, plants, animals, not happening in bacteria. All right, which is a good thing. That's really what allows genetic engineering to occur. We can feed bacteria DNA. We can feed and it will make the RNA that we want. It won't process it, it won't change it, and it will use it based on the DNA that we give it, that we feed it. All right, here's just, once again, more the same kind of pictures. Here's a piece of DNA with exons and introns. It's transcribed. It has the cap put on the 5' end, the poly A tail, the adenine put on the 3' end. That's the guanine, the GTP cap. The exons are kept, the introns are removed. So the splicing is that removing of the introns. Okay, well, if you're still watching, I guess it means you're either I'm making you watch in class or it's helping you. So I hope this is helping. Um, so yeah, we were looking at this picture of how introns are removed, exons are spliced together, and so yes, this mature mRNA is ready to be transported out of the nucleus into the cytoplasm for translation. Okay. Oh, sometimes you'll see these models. Same thing here, it's just more the same. DNA makes pre-mRNA, RNA is processed, mature mRNA floats out into the cytoplasm for translation. This processing, there is GTP, G with 3 phosphates added to an end. Sometimes you get these what are called UTRs or untranslated regions stuck in there. Hopefully you're familiar with what the start and stop codons are. Here's some more of that untranslated regions, and then a poly-A, a lot of adenine on the tail. Yeah, these caps, and it's just there to protect it. Really, this is the piece that's going to be used for transcription. This helps it make its journey, if you will, through the cytoplasm. Just helps it kind of maintain its integrity as it starts to touch and come into contact with everything else floating around in there. Okay, now we will spend a lot of time on this, and we'll do a few labs where we can turn on the promoter region of DNA. So really I just kind of wanted you to have this idea that out in front of the part, where transcription happens, kind of how the enzymes know to float in and make the right size bubble, or to make the bubble in the right spot, to make the fork in the right spot for transcription, depends on these promoter regions. So just think of the promoter region as just something out in front of where transcription is going to happen. But promoter region, promoter region. The promoter is a part of the DNA. The promoter is a region of DNA. That really helps as we move along. Okay, once again, another one of my favorite models. The thing I really like about this is you do kind of get that model of how, okay, here's how the pre-mRNA gets shortened and capped off. But the thing I really like about this is that a cell can take the same primary transcript, in other words, the same DNA was used, but make a different hormone, a different protein. So here a thyroid tissue making a hormone that regulates calcium. Here's brain tissue, brain cells, taking the same DNA, the same mRNA, processing it differently, and as a result, a different protein is translated. And this protein is functions as a pain receptor. So, I don't know, to me that's fascinating. Same DNA, same mRNA, pre-mRNA, different processing, as a result, different protein being made. Okay, so we've done kind of replication, you kind of understand some of the important aspects of transcription. Now let's look at the final piece, translation. And really, you've done a lot of translation, or at least played the letter game when you were using this chart. And I think we'll pick up here, because you guys really already do know what the wobble position is. Yeah, we got time. The wobble position is that third spot. So technically, it's the third spot on the tRNA that the mRNA sticks to. But you guys knew if I said, what can make valine? Well, GUU, GUC, GUA. I mean, GU is all you needed, and then that third position really didn't matter. And that's the case for most of these. That third position can be 2 different things, 4 different things. But yeah, the fact that the third position can change, that's what we call the wobble position. And it was actually discovered by Dr. Wobble, not the fact that it can wobble and change, but just kind of ironic that his name does also somewhat describe the properties of that piece. Okay, once again, I appreciate all your time. I hope this is helping. Okay, so yeah, really what we're looking at is translation now, the final piece of the central dogma. Translation is protein synthesis. We just briefly talked about that wobble position, that third spot that can vary. Okay, so the thing also what's important to remember about this is the universal codon amino acid chart. In other words, bacteria will use these same codons to make proteins. Human cells will use these sequences of nitrogen bases. These three nitrogen bases make a codon. So it applies for plants, animals, people. Every living thing uses or kind of uses the same code, if you will. So let's kind of talk about what does that really mean to use these three letters to allow an amino acid to make an amino acid. That just is kind of weird. Okay. Remember the difference is though our cells do this, but it happens where they want the protein. Prokaryotes are small. It happens right away. Even as the mRNA is being made, it will make this. So anyway, let's look at what is translation. Okay, so the mRNA will stick to what's called tRNA. So those kind of transcript things. So okay, here's a piece of mRNA, or here's a piece of mRNA. It will stick to tRNA, which here's a model of that. Usually on paper it's kind of drawn flat, but here is where the mRNA is going to stick, right there. Maybe right there, right there. Okay, so anyway, when it sticks to it, it's just naturally attracted to, those nitrogen bases naturally like to stick or bind. So here you can see the RNA is 5' to 3. It's sticking there. So GCC. GCC. You can go to here and find GCC, alanine, right there. So in fact, that is alanine is this amino acid stuck to that. So what's a tRNA? It's a nucleic acid in this funny shape, but at one end, an amino acid molecule is stuck to one end. And the thing is when this RNA sticks to it, this guy, that wobbles, that starts to, when something sticks to something, it changes it. It's going to affect all these side group interactions. This thing does start to move a little bit. It changes shape. It's denatured when that sticks. When this tRNA changes shape, maybe when this guy changes shape, boop, 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 the amino acid pops off. This guy changing shape causes this guy to go out of solution, and then if those pop off in the right order and enough energy is released from all the energy molecules that are in there, and the enzymes floating around in there help create it. But as these amino acids pop off, they bond together and form a protein. The order that they pop off is determined by these tRNAs floating in and sticking to the mRNA, sticking to the mRNA, sticking to it. Pop, 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 go the amino acids. Okay, so here's kind of a fancy way of looking at those steps from your textbook, different textbooks. Quite honestly, let's maybe put this in simpler terms. Okay, so remember, this is the wobble position right there. So not only could GCC make alanine, but also GCU was, GCA, GCG, GC, all of those would make alanine. And so that's the wobble. That spot there can be different things, but alanine would still be your amino acid attached up here. Alanine would still be your amino acid released out into solution. So yeah, so those steps of what I was talking about, the sticking, pop, pop, pop. Here's maybe a simple way to look at it. The amino acid sticks to the appropriate tRNA, then the mRNA sticks to the tRNA. This binding causes the tRNA to change shape. That causes the Once again, I cannot thank you enough for all your time. Sorry for the interruptions. Okay, we were kind of going over these simple steps, or the steps of translation, just trying to use simple language. So yeah, the amino acid sticks to the tRNA, the mRNA sticks to the tRNA. That binding causes the tRNA to change shape, to denature. That allows the amino acid to enter, to pop off. And then really as they come off in sequence, they are able to bond together. The mRNA, once it kind of goes through and all the tRNAs stick, it will leave. And then really the protein can then change after that amino acids are stuck together. The protein can undergo some more changes to its molecular shape too. And that's really our next chapter. We'll really just look all at proteins and how they're made and how they're modified and how they're used. Now the thing that, once again, I think is very, very worth mentioning is our central dogma. So here's maybe another way to look at it. The DNA makes the pre-mRNA, then mature mRNA, and then that's used to make a protein. But the thing that's important to remember here is what regulates each step? These enzymes. Enzymes even control, the whole process of opening up the DNA. Enzymes control this whole process of processing the mRNA and shortening it and removing introns. Enzymes affect this whole process of getting translation started. So enzymes are proteins. So the proteins that are made affect all parts of this process. And this last little piece I wanted to talk about or introduce to you is just this notion that, okay, sorry. So yeah, the last little piece I wanted to introduce was just this idea that, it's becoming very challenging to look at just a specific gene and follow its pathway through the cell because, everything that's in there affects everything else. And so right now we have a lot of sequencing data, and it can be correlated to health problems. It doesn't mean we know the cause. It doesn't mean we can model exactly how the gene is being transcribed or exactly how it's being processed or exactly how it's being translated or exactly how the protein's being modified. But we can find these mathematical correlations and then kind of go from there. So, but it is interesting to think about who do we want to keep this data? The government, or do we want private companies, or do we just want to give up on gene sequencing? I don't know. It's kind of fascinating debate currently going on. And so that's what the magazine's all about. Please, I hope you added this little interview to that magazine too. Okay, and then the final little piece I want to leave you with is just maybe going back to this central dogma, this just getting the DNA to start in the first place. Maybe it's how can we even get part of it transcribed in the first place? We briefly mentioned what promoter regions were, but maybe even just taking it, maybe even looking at it a little bit easier. Here's a scan of some DNA unraveling as it was all wound up in the chromosome. Here's a computer model of how that DNA is kind of, these blue are the histones or the proteins, and you can kind of see the double helix kind of all intertwined around that protein. In fact, that protein, that histone protein model, it kind of looks like our first, model, our first slide here, kind of the way we model proteins with those ribbon structures. But anyway, to get this DNA to get started, to get it unraveled in the first place, it's got to be exposed, and so it's got to stick out. That's where the things can touch. This part of the DNA that is exposed, we really say that it is, to say that it's exposed or allowing those enzymes to get access to it, to touch it, and to start the whole reaction. A lot of that looser DNA, the exposed DNA, we said it has acetyl groups added to it, or organic acids, carboxyl groups. They really can kind of cause that chromatin. Okay, thanks. I just almost finished. I just wanted to emphasize a couple of things. So I was trying to get you to really think about that to turn on a gene, to activate your DNA, the molecules, the enzymes, the energy molecules have to touch it. So it's one thing to diffuse into the nucleus. It's another thing to actually touch the DNA that's exposed. The last slide is just a computer model of that same kind of picture there, where you can see how it's being exposed. But yeah, the thing I wanted to go over with you here is to get them to touch. In chemistry, they maybe you learned this, but we just say things react because the molecules collide, and there's this what we call the collision model. And what that means is for molecules, they have to collide with enough energy, and secondly, they have to be properly aligned when they collide. And that's really what enzymes or catalysts do for reactions. Remember, enzymes are just catalyst in a cell, a biological catalyst. They're not really part of the reaction. They're not reactants. They're not products. But they set up an environment where these molecular collisions will occur with enough energy, and the molecules will be aligned properly. That's why I really like saying that enzymes set up or create an environment where this can happen. So enzymes can create an environment where this DNA is exposed and allow more enzymes, more energy molecules to get in and touch. Now, a couple things as far as loosening or allowing the DNA to be exposed. We can add these organic acids, these acetyl groups to it. They can cause the DNA to be exposed and really cause genes to be turned on. Now, turning them off. And once again, this is kind of a macroscopic level because we can actually see this. To turn them off, it's called imprinting of genes. So gene imprinting is turning off. Let me say that again. Gene imprinting is turning off. That also means you're adding these methyl groups. So these parts that are tightly coiled, that are in the center, that are all balled up there. Maybe if you look at the computer model, these parts up here that are all tightly coiled, still all wound up in their protein, those are turned off. It's fair to think most of those have been imprinted, most of those genes have been methylated. So once again, this idea of gene regulation is a big topic in this course and a big topic in biology. But right now you kind of have the idea that the DNA has to be exposed to be turned on. We can expose it by adding these acetyl groups to the chromatin, to that protein DNA complex. And the other way that we can regulate genes is we'll kind of look at what turns on those promoter regions. So these parts sticking out probably have a lot of their promoter regions need to be activated or turned on in order for that gene to be transcribed as well. Hey, thanks so much. If you made it through, I think you're in great shape for the quiz. Appreciate all your time.